Hi, I'm Naomi Moris from the Stem Cell and Human Development Lab. Our group is interested in figuring out how human embryonic development happens, how cells are able to differentiate into all the cell types of the body in a robust and spatiotemporally coordinated manner. In particular, we're interested in a process known as gastrulation that occurs as cells of the early embryo differentiate to form the three germ layers of the body, the endoderm, mesoderm, and ectoderm, organised along an anterior-posterior body axis. This process has been well studied using embryos from many various model organisms, including the mouse, but it's not possible to study in humans because of ethical limitations. And yet we know that human development can be quite different from mouse development in terms of the length of time that certain processes take and other details that can be quite strikingly different. There's also a need to understand the process from a specifically human perspective so that we gain a better understanding of what happens when the process goes wrong. Many congenital abnormalities or birth defects and miscarriages are thought to occur in early development as a result of genetic or cellular dysregulation. To try to dig deeper into how these processes are coordinated and can become dysregulated, we use pluripotent stem cells. Because these cells are able to self-renew indefinitely and can be differentiated in culture to many different cell types, we can easily use them in the lab to answer many questions. However, rather than growing them as flat two-dimensional adhesive cultures, we use three-dimensional methods to allow the cells to interact freely with one another. Under very defined conditions that we've developed, we can get these pluripotent stem cells to come together into a spherical aggregate, which breaks symmetry and undergoes axial elongation. Importantly, these structures are able to differentiate to cell types from all three germ layers that are organised spatiotemporally along an anterior-posterior axis that resembles the early mammalian embryo. We therefore call these structures gastruloids, and we use them as a model system to study the fundamental developmental biology principles of embryogenesis from a human perspective. I'm really excited to understand more about how human pluripotent cells interact with one another to generate coordinated patterns during early development, and I'm hoping to find a student who is just as keen to join us in answering these questions. If you think that's you, please do apply. You can find out more about the kinds of projects that will be available and our research on the Crick website, from our publications, and in the following short presentation. Thank you. In our group, we study the fundamental principles of human development using simple model systems. In particular, we use embryonic stem cells and induced pluripotent stem cells to understand fate decisions. We're interested to explore how coordination is achieved using these cells in three-dimensional aggregates known as gastruloids. We want to be able to use this simplified model system to interrogate properties of human development, including using them as models of congenital abnormalities and disease. Human gastruloids are made of many thousands of pluripotent stem cells aggregated in 3 dimensions. Over time, these spherical aggregates of cells are able to self-organize and display polarized gene expression in one region of the aggregate. One of the first genes to be polarized to one end is brachyury, or TBXT, which is known to be expressed in the primitive streak of the mammalian embryo. Over time, the gastruloids will begin to change shape and will elongate. They also start to express genes in particular regions along their length, which corresponds well to the anterior-posterior axis of the mouse embryo. It is for this reason that we think human gastruloids will be a good model of embryonic development. Using live imaging techniques, we can watch as the individual pluripotent cells come together into a spherical aggregate. In these movies, cells have 3 fluorescent reporters corresponding to expression of marker genes from the 3 germ layers, the endoderm, the mesoderm, and the ectoderm. You can see how expression of these genes appears to be spatially coordinated as the gastruloids develop, and that they become gradually localized to particular ends of the structure. Using RNA sequencing technologies, we've been able to further characterize the human gastruloids. In particular, a technique known as tomosequencing was performed in collaboration with the Hubrecht Institute, which let us examine the gene expression profile of genes along the whole anterior-posterior axis and revealed a surprising degree of organization. We're now interested in finding out how this organisation is established and how signalling gradients might be mediating the gene expression coordination. We're also interested in figuring out the underlying regulatory networks that are involved in setting up this process and keeping it so robust. Finally, we want to understand how specific embryonic processes occur, such as the process of somatogenesis, where somites are sequentially generated along the embryonic body axis. For instance, the dynamic timing of these processes can be very different between the mouse and the human embryo, and we're interested in finding out what controls some of these human-specific features. By developing these models, we're ultimately hoping to find ways to explore what happens during disease and developmental abnormalities. This might involve using patient cells or gene-edited cell lines to explore how dysregulation can lead to abnormal patterning and structural organization. Specific details about student projects in our lab will be defined following in-depth discussion with students so that we can develop a project of mutual interest. If you want to find out more, please don't hesitate to get in touch.